For years, we thought we had a pretty good handle on psoriasis, especially this key inflammatory player, IL-17. But get this, new research is showing us that the story is way more intricate than we ever imagined.
Yeah, it really is.
And that's actually thrilling because, well, it could pave the way for even better treatment.
Exactly. Today, we're taking a deep dive into the IL-17 family of signaling molecules, these things called cytokines, and their really crucial roles in psoriasis.
Okay.
Our main source for this exploration is a really insightful paper published in Frontiers in Immunology just last year, May 2023.
Right. Okay, let's unpack this. Our mission today is to really get a comprehensive picture of IL-17's involvement in psoriasis. We want to understand its central role, sure, but also dig into the surprising complexities that go beyond just the main player, IL-17A. Because while the IL-23T17, IL-17A pathway is definitely a major part of the puzzle, it's becoming clear it's not the complete picture.
Absolutely.
Yeah.
And by the end of our time together, you'll have a much clearer and hopefully more nuanced understanding of this really critical area of research.
Okay.
We're moving beyond the sort of simple version and getting into the fascinating details that are now coming to light.
So let's start with what we thought we knew. What's the classical understanding of IL-17A's role in driving psoriasis?
Well, the traditional view focuses on a pathway that kicks off with certain immune cells called dendritic cells.
Okay.
These cells release a signaling molecule called IL-23, which then activates a specific type of T cell known as Th17 lymphocytes.
Right.
And the main job we thought these Th17 cells had in psoriasis was producing IL-17A. You can kind of think of IL-23 as a general sending an order to the Th17 soldiers. And their main weapon in this battle was thought to be IL-17A.
Right, okay. And then IL-17A acts on the skin cells, the keratinocytes, is that right? What happens when it does that?
Precisely. IL-17A signals to the keratinocytes, leading them to produce even more IL-17. So it creates this kind of self-amplifying loop.
Ah, okay.
But it doesn't stop there. These keratinocytes also start releasing chemical signals that attract more inflammatory cells, especially neutrophils, further intensifying the inflammation.
So it's like pouring fuel on the fire.
Exactly.
Yeah.
It's like a snowball effect of inflammation.
And it's not just these Th17 cells making IL-17, is it? The paper points to some other cells involved.
That's a crucial point that challenges the older, simpler model. We now know that other types of immune cells, including CD8 plus T cells, also called TC17 cells,
TC17,
which actually reside right in the top layer of the skin, the epidermis, in psoriasis, can also produce IL-17A.
Right there on the skin.
Yeah. These cells have what's called a resident memory phenotype. That means they have a kind of memory of past inflammation and can quickly reactivate in the same spot.
Ah, contributing to flare-ups.
Exactly, contributing to those recurring flare-ups. Interestingly, they also express PD-1 and IL-23 receptors, so they can respond to signals in their immediate environment too.
Okay, so IL-17A is coming from multiple sources. What exactly does it do in the skin beyond just attracting more of those neutrophil inflammatory cells?
Well, IL-17A has a pretty wide range of effects. It stimulates the production of a whole host of inflammatory mediators, contributing to the redness, swelling, and pain we see in psoriasis. It also boosts the skin's defense against microbes by prompting the production of substances like cathelicidin and defensins.
Okay, those sound helpful usually.
They're usually helpful, for fighting off bacteria and fungi. But in psoriasis, this response can become overactive and actually contribute to the problem.
And it also messes with the normal function of the skin cells themselves, right? Like they don't mature properly.
Exactly. IL-17A disrupts the normal process of how keratinocytes mature and differentiate. This leads to a reduced expression of important molecules that are usually part of healthy protective skin.
Weakening the skin barrier.
Right. It weakens the skin's outer barrier, which is our first line of defense. Furthermore, it encourages the production of enzymes called metalloproteinase. These break down the tissue structure and can potentially make it easier for even more inflammatory cells to move into the skin.
Wow, it sounds like IL-17A is really orchestrating a major inflammatory event in the skin. And the paper even touches on indirect effects through these other signaling molecules, IL-19 and IL-24.
Yes, IL-17A doesn't act in isolation. It can also stimulate connective tissue cells in the deeper layer of the skin, the dermis. These are called dermal stromal cells to produce IL-19 and IL-24. These cytokines themselves can contribute to inflammation and the overgrowth of skin cells, adding another layer of complexity.
So it's not just direct effects on keratinocytes.
Not at all.
And finally, regarding IL-17A, it seems like it even interferes with the body's own attempts to regulate this inflammation, affecting regulatory T cells.
That's right. In psoriasis, regulatory T cells, which normally help to calm down immune responses, show a reduced ability to suppress inflammation. They can even become somewhat dysfunctional. Interestingly, this has been linked to changes in the release of a calming signal called transforming growth factor beta and an increase in an inflammatory signal, interferon gamma.
But there's some good news there.
Yes, the encouraging news is that treatments that block IL-17A have shown the potential to actually help restore the function of these regulatory T cells.
Okay, so IL-17A is clearly a big player, a huge player. But this is where the traditional sort of straightforward model starts to get challenged, right? It's not just about IL-23 leading to IL-17A from those Th17 cells.
Precisely. A really important discovery is that IL-17A can be produced by a variety of other immune cells independently of that initial IL-23 stimulation.
Independently, okay.
Yeah. This means that even if you successfully block IL-23, you might still have IL-17A being produced by these other sources.
Which could limit how well that treatment works for some people.
Potentially, yes, limiting the effectiveness of that approach in some individuals.
And who are these other independent producers of IL-17A that the paper highlights?
The paper points out several key cell types. Innate lymphoid cells, or ILCs.
Okay.
Gamma delta T cells, invariant natural killer T cells. Mucosal-associated invariant T cells, a bit of a mouthful, so MAI T cells is easier.
MAT cells, got it.
And even neutrophils themselves are capable of producing IL-17A without needing that initial IL-23 signal.
Wow, okay, so there are multiple pathways leading to IL-17A production.
That's the key takeaway.
That definitely makes things more complicated. So moving beyond just IL-17A, the paper emphasizes that other members of the IL-17 family have been somewhat overlooked. Can you give us a quick overview of this whole family?
Sure. The IL-17 cytokine family includes 6 main members, labeled IL-17A through F.
A through F.
Right. What's interesting is that IL-17A, B, C, D, and F can form active homodimers. Think of it like these molecules pairing up with identical twins to become functional.
Okay.
Additionally, IL-17A can also form a heterodimer by pairing up with IL-17F, like a fraternal twin.
A and F together.
Exactly. These cytokines then exert their effects by binding to a family of 5 related receptor subunits named IL-17RA through E on the surface of cells.
Okay, so it's a whole family of related signaling molecules that can team up in different ways. Let's zoom in on IL-17F first. The paper mentions it's actually more common than IL-17A in psoriatic skin lesions. That's quite a surprise.
It is a surprising finding, yeah. And it suggests that IL-17F might play a more significant role than we previously thought. And while IL-17F has similar functions to IL-17A, particularly in the body's defense against infections, it's generally considered to be less potent.
Less powerful.
Less powerful, generally.
Right.
Interestingly, the IL-17AF heterodimer, the fraternal twin pair,
Right.
it has a level of potency that falls somewhere in between the IL-17A and IL-17F homodimers.
Okay. Intermediate potency. And where is IL-17F coming from? Is it produced by the same cells that make IL-17A?
Well, initially the assumption was that IL-17F was generally produced alongside IL-17A. Made sense.
Yeah.
However, recent analysis of immune cells taken from psoriasis patients has really challenged this idea.
How so?
It turns out that the majority of Th17 cells in psoriasis actually express either IL-17A or IL-17F.
Either of both.
Not usually both, no. Less than 10% seem to produce both simultaneously. In fact, within the skin lesions themselves, a larger proportion of T cells seem to be expressing IL-17F rather than IL-17A.
Wow.
So the old idea of a single Th17 cell pumping out both might be wrong, meaning treatments only hitting IL-17A could be missing a significant part of the picture.
That's a really significant finding with direct implications for treatment. It suggests that just blocking IL-17A might not be enough in some cases, right?
Exactly. This likely helps explain why therapies that target both IL-17A and IL-17F, like bimekizumab, have demonstrated superior effectiveness in clinical trials.
Especially for people who didn't respond well before.
Precisely. Especially for individuals who haven't responded adequately to treatments that only block IL-17A. It also raises the possibility that IL-17F might contribute to the recurrence of psoriasis lesions after treatment.
And maybe psoriatic arthritis, too.
Its role there is still under investigation, a bit debated, but some studies have found increased levels of both IL-17A and F in the joint tissue of affected patients. So, potentially.
Okay, next up is IL-17C. This one sounds particularly interesting because it's mainly produced by and acts on the skin cells themselves, the keratinocytes.
Yes, IL-17C is unique in that sense. It only shares about, 23% similarity in its structure to IL-17A.
Quite different then.
Yeah, quite different. It signals through a receptor made up of IL-17RA and IL-17RE subunits. Physiologically, its normal job is to help establish those important protective antimicrobial responses in barrier tissues like the skin.
But in psoriasis, it seems to be involved in the early stages of inflammation and the development of those characteristic skin plaques.
That's a key insight from recent research, yes. RNA sequencing studies suggest that IL-17C acts as a crucial regulator of the initial network of signaling molecules.
The kickoff crew.
Sort of, yeah.
Yeah.
Involved in the development of psoriatic plaques, it appears to be involved in setting the stage for the full-blown inflammatory response in the skin.
And clinically useful, too.
Potentially very useful. The levels of IL-17C in the affected skin decrease significantly following successful treatment with various effective therapies, including those that block both IL-17A and F, traditional treatments like methotrexate, and even ustekinumab.
And in the blood, too.
The same pattern has been observed in blood samples, yeah, which suggests that IL-17C could potentially serve as a biomarker.
A way to track treatment success.
Exactly, to help monitor how well a treatment is working.
So tracking IL-17C levels could give doctors a way to assess treatment effectiveness. What about the other members of the family, IL-17B, D, and E? Do they play significant roles in psoriasis?
Well, IL-17B primarily signals to the IL-17RB subunit and is more strongly associated with roles in cancer.
Okay, mostly cancer.
Yeah. In psoriasis, its concentration in the skin plaques appears to be quite low, suggesting it doesn't have a major role in the main inflammatory processes of the disease.
And IL-17D?
IL-17D is the least understood member of the family, really. Its specific receptor hasn't even been identified yet.
Still a mystery.
Pretty much. It seems to be involved in anti-tumor and antiviral responses. Interestingly, its levels are lower in typical chronic plaque psoriasis, but are elevated in a specific type called palmoplantar pustulosis.
The one affecting palms and soles.
Right. And there, it might contribute to inflammation mediated by another signaling pathway involving the IL-36 receptor, so maybe a role in specific types.
And IL-17E, also known as IL-25, it was initially linked to type 2 immune responses like allergies, but it sounds like it also has a connection to psoriasis.
That's correct. While primarily known for its role in type 2 immunity, IL-17E can also contribute to skin inflammation in an autocrine manner.
Meaning it acts on the same cells that make it.
Exactly. It can act on the same keratinocytes that produce it. It can induce the expression of pro-inflammatory cytokines, further amplifying the inflammatory network in psoriasis and promoting the recruitment of neutrophils.
So it adds to the fire, too.
It does. Interestingly, it has unique effects on keratinocytes, influencing their ability to move and multiply, suggesting maybe a broader role in the skin's overall balance, its homeostasis, beyond just being an alarm signal.
And potentially another biomarker.
Possibly. Elevated levels of IL-17E in the blood might even serve as a biomarker for psoriasis activity, particularly in severe forms like erythrodermic psoriasis, which affects the whole body surface.
Okay, and how does it signal?
It signals through a receptor composed of both IL-17RA and IL-17RB subunits, which again highlights the interconnectedness of the family's signaling mechanisms. You see RA popping up again.
It really is a complex family affair. So this whole family of IL-17 signaling molecules is clearly much more interconnected and complex than we initially realized. How has this deeper understanding translated into actual treatments for psoriasis that are available now?
Well, the success we've seen in treating psoriasis by targeting the IL-17 pathway has been truly remarkable. We now have highly effective biologic medications and also JK inhibitors that specifically interfere with the function of IL-17 and related signaling molecules.
Right, and the paper gives a good overview of some of these drugs. Can you highlight a few key examples and what their specific targets are?
Certainly. We have medications like secukinumab and ixekizumab that specifically block just IL-17A.
The original target.
The original target, yes. Then there's bimekizumab, which takes that broader approach and targets both IL-17A and IL-17F.
Covering both bases we talked about.
Exactly. Then there's brodalumab, which targets the IL-17RA receptor subunit.
Ah, the shared receptor part.
Right. So because RA is part of the receptor for several IL-17 family members, blocking it effectively blocks signaling from multiple IL-17 cytokines.
Okay. And upstream.
Upstream, yes. We have ustekinumab, which targets the P40 subunit shared by IL-12 and IL-23, and risankizumab, which is more specific and targets the P19 subunit of just IL-23.
Cutting off the signal earlier.
Exactly, indirectly reducing the production of IL-17 family members. And finally, we have GK inhibitors like tafasitamab, which is quite broad, and deucravacitinib, which specifically inhibits TYK2, a member of the JK family. These broadly dampen down various cytokine signaling pathways, including those involving IL-17.
It's interesting that the paper points out that blocking IL-17A seems to lead to a quicker improvement in symptoms compared to blocking IL-23 further upstream. Why might that be the case?
That's likely because directly blocking IL-17A targets a key downstream inflammatory mediator that's actively driving the disease right there at the skin level.
It's closer to the action.
Exactly. On the other hand, blocking IL-23 works further upstream by trying to reduce the production of IL-17A. It's sort of like going straight to put out the fire versus trying to turn off the signal telling the firefighters to go there in the first place.
Okay, that makes sense. But then why might inhibiting IL-23 have a potentially more long-lasting therapeutic effect, as the paper suggests? That seems a bit counterintuitive.
Yeah, initially the thought was simply that it takes time for the Th17 cell population to kind of rebound after IL-23 is blocked. Makes sense.
Right.
However, more recent research suggests that IL-23 inhibition might also lead to a reduction in those resident memory T cells in the skin.
The ones that stick around and cause flares.
Exactly, the ones we talked about earlier. These cells contribute to the chronic nature and the recurrence of psoriasis. So, reducing their numbers could potentially lead to a more sustained remission, a longer-lasting effect.
It really sounds like we're still uncovering the nuances of this intricate network. And the paper hints at ongoing research and the development of even more targeted molecules within the IL-17 family.
Absolutely. This is a very active area of research. Scientists are constantly exploring new ways to precisely target different members of the IL-17 family and their receptors.
Aiming for more precision.
Right. The goal is to achieve even better and perhaps more personalized treatments for psoriasis with fewer side effects in the future.
So to bring it all together then, it's clear that the role of the IL-17 family in psoriasis is far more complex, much more interconnected than we initially understood. While IL-17A is undoubtedly a central player,
Definitely central.
IL-17F and IL-17C seem to have significant and distinct contributions, particularly in terms of treatment response and those very early stages of inflammation. And while the IL-23 Th17 IL-17A axis is certainly important, it just doesn't tell the complete story of this disease.
Precisely. And this deeper understanding is so valuable because it explains why treatments that target different points in this pathway, including multiple members of the IL-17 family, are showing such great promise in improving the lives of people with psoriasis.
Makes sense of the newer drugs.
It does. It also underscores the critical importance of ongoing research to unlock even more effective and hopefully tailored therapies that address the individual complexities of this condition.
It really makes you think, doesn't it? Given this intricate web of signaling molecules and the emerging roles of these different IL-17 family members, what other potential therapeutic targets within this network might hold the key to even better, maybe more personalized treatments for psoriasis down the line?
That's the big question, isn't it?
It's definitely a fascinating and rapidly evolving area to keep an eye on.